Good afternoon, ladies and gentlemen. Thank you for staying almost until the end of the session. My name is Boris. I'm a PhD candidate from National University. And today I'm gonna share our discoveries in the work we have been developing for the last 3 years or so. An automated method for the modeling of bio-crudes from GCMS and elemental analysis experimental data. Now, after introduction, in which I will describe why are we doing this and process simulation challenges, I will compare our methodology to the current trends, and in materials and methods I will explain in a bit more detail the reduction method and the efforts we have done to automate it by software, as well as the minimum experimental data input, after which you will see results and I will conclude. Now, first and foremost, why are we doing this? Well, as you may know, the biofuel research, especially over the last 10 years, has been intensifying as the global energy policies are shifting towards renewable energy technologies. Now, researchers have researched everywhere from first to third generation biofuels, ranging from things like food crops, sawdust, rice husks, switchgrass, and particularly here in Korea, third generation biomass seaweeds. Now, as these are all very different things, they all have something in common when subjected to thermochemical conversion, especially conversions which yield liquid products. That is, that they yield the so-called bio-crudes, which is this black viscous liquid. Which are extremely complex organic mixtures containing up to hundreds of compounds, mostly oxygenated hydrocarbons. Now, it is all well and good on experimental and lab scale. It's simply straightforward producing a bio-crude. But simulating this in process design, in a simulation package, in order to assess the techno-economical feasibility of a possible production process, is a very different thing. And herein is the main challenge, and the reason for this method is how to model these complex organic bio-crudes with maximum simplicity and accuracy. Now, there are many approaches currently used for this, and these four are the most common ones. You can model the decomposition kinetics using key biopolymer components, and this approach has some predictive capabilities. However, it does not have minimization of components inside the method, and it does not utilize experimental data completely. Now, points 2 and 3 are essentially the same, that is utilizing a set of pseudo-components, or in process simulation lingo, assays. The only difference being either you're using only pseudo-components or some combination of pseudo-components and real components. And I might add that this method is utilized by United States National Renewable Energy Laboratory, and they are in this field usually an example which we all look upon to. Now, this is essentially manually mixing and matching to tune the properties of the mixture, and especially true boiling point curve is an objective function here. This is relatively quick, and the true boiling point curve is good for modeling things like distillation columns. However, there is definitely a reduction of accuracy the more pseudo-components you use, and also there is a large number of components needed, 40 and upwards. And finally, you can manually pick components from your experimental data set and compare them with your simulator's data set. Now, this is closest to the true bio-crude mixture if you do it really carefully. However, this does require some a priori knowledge of the experimental mixture, and if you're doing it by hand, it can really take a while. And if you have a lot of data sets, that's quite a while. Now, the novelty of our method here, first and foremost, that it is automated, which obviously means fast execution, and as an objective function, it has the minimization of the number of components. Which reduces time and complexity of process simulation, which I will demonstrate later. We have tested it so far in 11 different data sets, and we are confident that the applicability is wide upon various feedstocks. It is user-friendly, requiring only your data set, and input and output are through Excel spreadsheets. It provides unbiased and accurate representations. It is almost completely based on direct utilization of your experimental data. And the output is ready to use for any process simulator you might be using. Now, you remember I said that the method has the minimization of components as an objective function. Now why is that? So, the Bio-crude, being complex mixture, it's a real challenge to accurately simulate in, for example, what we used Aspen Plus. You have to take into account that there are water non-soluble components, semi-soluble or completely insoluble components, all in this very unclear phase transition. So to simulate this complex mixture, the straightforward way is to utilize the so-called activity coefficient method, or NRTL, whoever is familiar with Aspen. And this method, as activity coefficient based, it predicts vapor-liquid and liquid-liquid equilibriums. And for the calculation of the liquid coefficient, it required the set of experimental or predicted binary parameters. Now, at this point I would like to mention it is very useful to utilize the components which are already in the simulator's database, because there is a great probability that these binary parameters are from some experimental data set and they are to be trusted. If not, in case of Aspen, you can predict them based on some correlations, but it is always useful to use components which are very well described within the database. Now, as you may see in the example of 5 components, the number of required binary pairs is 10, and that number rises geometrically as you increase the number of components. So now you might see in some normal data set straight from the lab, you have, let's say, 150 components. You are over 5,000 region. Now, depending on your hardware capabilities, unless you have a supercomputer, that it will take you most of a couple of days. Now, this is one of the main reasons why we have put the minimization of components as our objective function. Now, the reduction method itself, many people ask me, because GCMS is not quantitative in nature, and I agree. That's why the reduction method is essentially the combination of the GCMS data with 3-level grouping and another combination of very strict reduction criteria. This is essentially how it works. For a given set of data, first the chemical grouping is applied, where you group your data into chemical groups, aliphatic, aromatic, and so on. Once you have the first grouping, then these chemical groups are grouped based on user-inputted critical retention time. Now, this will group the components close in molecular mass and boiling point within the same chemical group. The third grouping is atomic, well, for the lack of a better word, sub-grouping, which will group components which have same atoms in their molecular formula. For example, if component G has this grouping, then it's aromatic from 0 to 5 minutes in chromatograph, and it has one of these combinations. Now, after this grouping, then the reduction starts. After the final grouping, the compound in this subgroup, which has the greatest normalized peak area, is remained within the subgroup, while other components are eliminated, and their values of normalized area are added to the first component, thus increasing its relative abundance, but keeping the ratio of those abundances similar to the original mixture. Now, therein comes the third reduction criteria, which is essentially testing of the reduced mixture against the original mixture, where the weighted average molecular masses and boiling points are measured for chemical groups of the reduced mixture, and the absolute difference between the same things in the original one is calculated. If that difference is above the reduction criteria, then the reduction is not well performed. The criteria was for molecular mass 15 grams per mole and for boiling point 30 degrees Celsius. Now, as you might notice, doing this by hand, which I did personally, it takes about 2 hours when you're experienced, even more when you're not. And when you have lots of data sets, it will take you most of the day, if not more. That's why we have developed a software which will automate this procedure and add a couple of more things to make it even more useful. The software is divided into 3 sections. The first one doing what I've just explained, the reduction method, after which it performs the compound database of chosen process simulator cross-referencing, which will actually find and replace components which are the most similar, so that the output has only components from the process simulator database. And you will remember why this is important from the previous slide. In the end, it compares with experimental data and performs reactor balance by keeping the reduced mixture fixed and slightly varying the composition of solid and gaseous product within some normal experimental ranges. Now, we have tested this thoroughly and validated against process simulation on 7 different data sets. And at this point, I would like to note that the more experimental data you have, the better. The simulator will have a better starting point for optimization. However, minimum data input is the GCMS of the Bio-crude, the phase distribution or yields of your gas, liquid, and solid, and initial guess for gas and biochar compositions, which you can get from literature or just make an educated guess, after which it will vary them to close the reactor balance. Now, how to validate if this is actually useful for a process simulator? We have used the reduced mixtures from 7 very well experimentally described data sets in a typical process simulation example to model industrial scale pyrolysis. Now, this is what you might do in a lab, but we simulate it on industrial scale, so you do have a thermochemical conversion unit. It might be a fixed bed reactor, a fluidized bed reactor, whatever. After which you separate the solids from pyrolysis vapors, and pyrolysis vapors need to be first liquefied and quenched, and the Bio-crude needs to be dewatered in a distillation or quench column, and non-condensable gases needs to be also separated. Now, if this method is valid, then obviously the reactor balance needs to be absolutely correct. The composition of biochar, the non-condensable gases, has to be at least within some reasonable point with experimental data. Obviously the yields need to be exactly as experimental data. And also, the biggest test is, does the chosen property method, which was NRT-LRK, works for this distillation column, and does the properties of the Bio-crude, chosen ones, accurately represent the actual experimental Bio-crude. As I mentioned before, this was tested on 7 different data sets. The best described we have were all from pyrolysis of seaweed, but at different reactor conditions and different reactors. As you can see here, the experimentally found compounds, and these ones are only for organic phase, not for aquatic phase, were these, and reduced mixtures, these were the total numbers of compounds, and the reduction ratio, which is the ratio between this number and this number, is an average 3.5. So the total number of compounds was on average reduced by the factor of 3.5, and it took, well, a normal desktop computer about 3 minutes per data set. As you can see, we have compared the yields of gaseous, aqueous, and organic phase, and also the chemical composition of the Bio-crude, experimental and simulated, and you can see that the results are indeed statistically significant. The other factors we have compared are as follows: energy recovery, calculated like so, mass yield, the heating value, and the water content of the dewatered Bio-crude. And as expected here, all the parameters of the simulated bio-crude containing only 10 to 15 compounds and original bio-crude containing 100 and more are within strong statistical significance. Now, to better represent this graphically, this is actually what is happening. If we plot normalized peak areas, which we have from our experimental data, against retention times, the green ones is the actual organic mixture with n number of compounds and n values of normalized peak areas, whereas orange dash line is the reduced mixture with less number of compounds but increased normalized peak areas, and the ratio of chemical groups remaining the same. And as you can see here, when you plot cumulative peak area against retention time, how well does the simulated data, or how unwell, fit the organic mixture. We could only do this point for data sets from other feedstocks which we have found from literature, and as you can see here, in case of seaweed fluidized bed pyrolysis, fixed bed pyrolysis, sawdust, and rice husk, the total number of compounds ranging from 23 to 127 was reduced to 6 raised to 25, and the potential number of binary parameters was reduced by a factor of ranging from 17 to 26.7. And in conclusion, in this work, for the first time, we have developed an automated modeling method for modeling of bio-crudes and inputting the ready-to-use results in a process simulator. And being tested on 11 datasets so far, and requiring 3.5 minutes per dataset, we think we have established a wide applicability and also fast execution, as we demonstrated. The validation was performed using an Aspirin Plus process simulator, and it has shown that this method produces reduced mixtures which represent complex mixtures with statistically calculated accuracy based on parameters which are usually valued in biofuel process simulation. And at this point, sorry, how are we on time?
Running out.
All right. At this point, I would like to acknowledge the National Research Foundation of Korea and also Energy Technology Evaluation and Planning and the Ministry of Trade, which were the main financiers of this project. And a very special thanks to my co-authors, Dr. Žuvela, Dr. Liu, and Dr. Jae-Hyun Choi, without whose help and input we couldn't publish this work in Applied Energy this year, and also patent pending for the method in both US and Korean markets. Well, thank you very much for your attention.